Good morning. Hi, everyone. So welcome to Friday Lecture. Today we're going to be talking about biological polymers. Those of you that might have read ahead in Chapter 5 know that one of the molecules we're going to be talking about is actually not a polymer, right? And so we'll talk about that. But these are basically all molecules that are very large, tend to be very large molecules called macromolecules in the cell. And it's important that before we really dive into what's inside of cells and how they work, we have to understand the molecules that are going to be playing the key roles in those cells. And that's what we want to do today, is really get down the basis for those molecules. Now, one clarification from last time, we talked about atomic number. You know that I'm sure all of you know this, atomic number is number of protons in the nucleus, right? Not the number of electrons. It could be the number of electrons. It could correspond to the number of electrons for a neutral atom, but it's defined by the number of protons. Just want to make sure that's clear. Okay. So today we're going to talk about these four classes of molecules. And this is all material that's found in Chapter 5. I really encourage you to read all of Chapter 5. There's a lot of really interesting stuff in there that gives you some of the interesting stories behind the discovery of these molecules and their behavior in the cell. And we'll try to touch on the highlights today, but there's a lot more that you'll find in Chapter 5 that will really flesh out this information. Okay, so basically all living things are made up of, really, it's kind of remarkable, just four classes of big molecules. And these are molecules that are called either carbohydrates, lipids, proteins, and nucleic acids. And in all cases, as we'll see, these are molecules that are made from building blocks that are much smaller. So small organic molecules that are linked together to make much bigger compounds. And so this is very important to know. Building blocks of the cell, sugars, these can also be called monosaccharides or carbohydrates, right? These are the building blocks of polysaccharides, very large polymers that we'll talk a little bit about today and more later on. Fatty acids are the building blocks of lipids and membranes, so very critical for life, right? These are the protection for the cell. They allow the cell to sequester material inside from the outside environment. Thirdly, amino acids. These are the building blocks of proteins, real workhorses of the cell. They do all sorts of things. They form enzymes. We'll talk more about that in a later lecture. And then nucleotides, which are the building blocks of nucleic acids, DNA and RNA, the genetic material. All right, so we're going to talk about each of these four classes in a little bit more detail. And I want to start off by talking about carbohydrates. So carbohydrates are used in cells both as fuel, and they're also used as building material. Incredibly important for plant cell walls, for example, for forming the outside of insects in a compound called chitin that forms the shells of things like cockroach outer shell and other bugs like that. And the simplest carbohydrates are called monosaccharides. These are single sugars. And to make carbohydrate macromolecules, these single sugars come together in much larger polymer chains. All right? And let's see if there's something else here I was going to tell you. Right, so does everybody know the definition of a polymer, right, is simply a long molecule that it consists of either identical or very similar building blocks that are linked covalently, right? That's the definition of a polymer. So if we look at the chemical formula for sugars, for monosaccharides, these all have molecular formulas that are multiples of this, CH2O. And so it's kind of easy to remember this, right? Because the word carbohydrate, there's the carbon, and there's the water, right? So carbohydrate. So that's a formula that tells us that these are molecules that consist primarily of carbon, hydrogen, and oxygen. And occasionally we'll find a nitrogen in there for special kinds of sugars. But in general, this is the formula that we'll see for monosaccharides. Glucose, and the molecular formula is here, C6H12O6. This is by far the most common monosaccharide in biology. It's the fuel for our brain, right? We have to have glucose, and it's used for all sorts of other things too. And as you can imagine, glucose is a very important building block for larger polysaccharides and other kinds of carbohydrates. And we'll see a little bit of that today, and we'll talk more about it later. Monosaccharides are classified by a couple of things. One is the location of the carbonyl group, and this is either, and we'll see this on the next slide, but the carbonyl group is going to be either in the form of an aldehyde, in which case in a sugar it's called an aldose, with the ose ending, or it's a ketone, in which case it's called in the form of a sugar, it's called a ketose. So in general, when you see the ose, O-S-E, when you see that terminus on a word, you'll know that we're talking about some kind of a sugar, right? So for example, if you see the word pentose, that tells us that it's a 5-carbon sugar, okay? And as we said, monosaccharides serve as a major fuel source for cells and also as the raw material for building much larger polysaccharides of many different flavors that turn out to be critically important for storing energy in the cell and for helping the cell have its shape and for organisms to have certain structural elements that are critical for function. Okay, so this gets into a little bit of the structure of sugars, and there's a couple things. So when you read your book, you're going to see that we can actually write out the structure of a sugar in a couple of different ways. We can write it out as a linear molecule, and these are sugars have somewhere between 3 and 7 carbons in them, right? They can have 3 to 7 carbons, and we can write that out as a linear molecule. And in your book, you'll see they write it as a chain of carbons that are linked together with OH and H groups coming off the sides. But it turns out that in aqueous solution, so in water, and we know from last time that basically all of our cells are in an aqueous, at least the inside is an aqueous environment, right? These sugars are actually cyclized, right? And so the typical way that we write them, which is sort of more chemically accurate, is to write them in this ring form that's shown here. And so here's the structure of glucose, for example. And one thing to note, it's sort of important in terms of the chemical structure of polysaccharides, is that the position of the hydroxyl group here on this ring, this hydroxyl group could either be pointing down in terms of with respect to the position of this group here, or it can be pointing up, right? So they're showing it to you in this configuration here with it pointing down, right? So this is the structure of alpha glucose, and if that hydrogen, that OH hydroxyl group were pointing up and on the same side of the ring as this group here, then we would call this a beta glucose. Okay, so that's seems like a detail, but it's actually really important because the chemical behavior of those glucose building blocks is different, and the polymers that they form are different, and we'll talk a little bit about that as well. Okay, but I want you to be aware of that, and your book talks about it too. So it's very important to understand a little bit about how these individual monosaccharide building blocks can come together to form polymers, and the way they do this is through a reaction called a dehydration reaction. It's also called a condensation reaction. And you can see here that what happens in this type of reaction is that we're losing a water molecule in the course of linking together these two monosaccharides through a covalent bond. And so this shows the reaction of glucose, and as we said, this is an alpha glucose, with a second monosaccharide called fructose. And you may know that fructose is actually the sugar that's found in fruit, right? So hence the name, fructose. And when these get linked together, we form a disaccharide called sucrose. And this is just table sugar. So sucrose is actually a disaccharide of glucose and fructose. And this is the linkage, the covalent linkage that occurs. It's called a glycosidic bond, or a glycosidic linkage. And in this case, we're forming a glycosidic bond between the carbon that's labeled carbon number 1 in this ring on the glucose, and the carbon that's labeled carbon number 2 on this ring, which is the fructose. So this is called a 1,2 glycosidic bond, or glycosidic linkage. And you'll see that in your book they talk about other kinds of disaccharides. For example, they talk about maltose, which has a different kind of linkage here. And because of that, it has a different kind of chemical property. So the reason that we detect sucrose as a very sweet compound is that it interacts with receptors that we have on our tongues that can detect this compound and break it down. Other kinds of disaccharides, even though they might look somewhat similar chemically, have a different ability to interact with our receptors and our enzymes, and so we might not taste them as sweet. Okay? Okay, and then polysaccharides can be huge, right? So these can be these can have many individual monosaccharide building blocks that come together to form these very long polymers. And a couple of examples are shown here. These are very abundant in biology and really important to know a little bit about. On the left-hand panel, this is a picture of starch, which is a polysaccharide that's found in plants. And they show you a couple of different examples here. These are found in actual organelles inside the plant cell. And these are, they don't show you this here, but these are actually alpha glucose units that are linked together by a 1,4 glycosidic bond. So a little bit different than what we saw in the previous slide, right? And here's something I think really interesting. So cellulose is a polysaccharide that forms cell walls, and we've heard a lot about it in the popular media because there's a lot of interest in using cellulose or some cellulose breakdown products as biofuels and for biofuel production. And one of the real challenges about cellulose is that it's very hard to break it down, and this is true across the biosphere, right? So one reason that plants use it is that it's really tough to break it down, and that means that insects don't have a very easy time breaking down cellulose, and neither do animals or humans. And so the interesting thing about cellulose is that it's a compound it's a polymer that's not that different from starch, except that it's a polymer of 1,4 linked beta glucose units, whereas starch is a polymer of 1,4 linked alpha glucose units, right? So it doesn't sound very different. In fact, the chemical formula would be the same, but the way they're put together is different. And so that difference, it sounds subtle, but it has a huge effect on the ability of enzymes to break down those different kinds of polymers. We can break down starch pretty easily. We can't break down cellulose without a lot of difficulty, okay? Okay, so let's say a little bit about lipids. So this is the second class of macromolecule that I wanted to introduce today. And these are molecules, although they can be very large, they're actually not polymers. And the sort of the unifying property of all lipids is that they don't like water, right? They tend to want to avoid water, avoid interaction with water. And that's a property that, in fact, is incredibly useful in biology, particularly for forming membranes that allow cells to segregate what's inside the cell from what's outside the cell and control what's getting into and out of the cell. Why do they not like water? Why are they called hydrophobic, right? That's the word that we typically use, hydrophobic, water hating. They don't like water because they consist mostly of hydrocarbons. That means carbon and hydrogen units together. And these form nonpolar covalent bonds, and so they really tend to not have the ability to hydrogen bond with water. Remember last time we talked about the fact that water molecules hydrogen bond with each other, and they also hydrogen bond with other molecules. But with lipids, that doesn't occur very easily, right? And so these two kinds of molecules tend to want to avoid each other. And we want to say a little bit today about the three of the most biologically important lipids, which are fats, phospholipids, and a class of molecules that we hear a lot about in the news called steroids. So what are fats? So these are constructed from two kinds of smaller molecules. one called glycerol and one called fatty acid. So glycerol is a 3-carbon alcohol, and it has a hydroxyl group attached to each carbon. And I thought I didn't put this explicitly in the slides, but we can write this out. So this is sort of an example of the linear way that we could write out a sugar. Okay, did I do that right? So that's glycerol, right? And this is actually important to know, because if you know the structure of glycerol, then you sort of inherently, as long as you understand how fatty acids can add on to these covalently, then you will know the structure of a class of compounds called triglycerides that are very important biologically. So this is glycerol. Okay, and then we can also draw out the structure of a fatty acid. So a fatty acid is just a carboxyl group that's linked to a chain of methylene groups or methyl groups, right? Or CH2, making a long chain and ending in. Okay, so that's a fatty acid. And this chain length could be of different lengths, could be branched. Sometimes there are branched chain fatty acids. I'm just showing you one with a straight chain. And if we know the basic structures of these two kinds of molecules, then we can easily figure out how a triglyceride is put together, because they're put together just by an esterification reaction that occurs between this fatty acid and one of these hydroxyl groups on the glycerol. Right? And so if you go to the next slide, I like this figure a lot because, and this is from your book, but this shows here's the glycerol unit right here, except now it's been esterified with three individual fatty acid chains, right? So you can see those here, and they show you sort of a space-filling model of that right here. So it looks very straight, very linear. And there's a couple things that are really important about these types of molecules. So first of all, most animal fats actually look like this. So fats that are, of this type that are made in animal cells tend to have what are called saturated fatty acid chains. That means that all the carbons in that chain have the maximum number of hydrogen atoms that they could have, right? So it's called a saturated chain. And the consequence of that is that these chains tend to be very straight, like this, like shown right here, or like this picture here. And they give you an example of butter, which contains a fat called stearic acid. And this is a saturated fatty acid. And so there's an interesting chemical consequence of this structure, which is that these chains can pack together very closely because they are linear, right? So they can pack closely together, and that means that at room temperature, these compounds tend to be solids, right? So butter tends to be a solid at room temperature, and that has consequences for the way that these compounds will behave in our bodies when we consume these compounds, right? And so you probably know that saturated fatty acids tend to be associated with a process called atherosclerosis, which is the buildup of fats on the inside linings of blood vessels. And part of the reason for that is the chemistry. These are compounds that have a structure that causes them to be solids at lower temperatures. And in contrast, here's an example of a plant, in this case an oil, something that is an oil at room temperature, olive oil. And in plants, fatty acids tend to have unsaturated carbon chains. And by that, what we mean is that they have one or more double bonds, like the one shown right here, which means that they don't have the maximum number of hydrogen atoms in the chain, right? So they're unsaturated. And there's another very important property of plant fats, which is that these unsaturated sites tend to have cis linkages. And I want to just show you that. You might remember this from chemistry class, but in case you don't. Right, so we can have a fatty acid that has a double bond in it, and we can look at, and so there's going to be a hydrogen here and a hydrogen here, right, and these are CH2 groups here, and we can look at the configuration around that double bond, and if it looks like this, the way I've drawn it here, this would be called, does anybody know? What kind of a configuration is that? Cis, right? Okay, right. So I think a lot of people know this. So this is a cis configuration, and this is typically what's found in plant fats. They occur naturally. And there's an important consequence to that. Why do we care what the configuration is? Well, we care actually because it affects the geometry of those fatty acid chains. And so in the slide, what you can see is that when you have a cis linkage, you get a chain that can bend in the middle around that cis linkage. And because of that, those chains are no longer straight. And so those fats no longer pack together as efficiently. And that means that at room temperature, an oil like olive oil, plant oil, will be a liquid. Okay? And so it tends to be a healthier fat because it tends to accumulate less in our bodies, in our blood vessels, and doesn't lead to the kind of atherosclerotic plaques that we observe with animal fats like butter. And then, of course, if we have a cis linkage, you can imagine that there's another way to configure this, right? And then probably this is sort of obvious, but we could also have a configuration that looks like this, where the hydrogens are now coming off in opposite directions and the chains are coming off in opposite directions, and this would be a trans linkage. And so this is important because, you know, when you go to the grocery store and you are trying to decide what type of cookies to buy, and you are looking on the nutritional label and it says no trans fats. What does that mean? It's one of those things. And now you know what it means. It means that it doesn't have this type of triglyceride fat present in those cookies. And why is that important? Well, it turns out that a lot of baking companies will hydrogenate fats that start off as plant fats, for example, and they will do this so that they are solid at room temperature. Why do they do that? Well, it makes it easier for baking, and it makes them have a longer shelf life in the grocery store. So from the food corporation's perspective, that's a good thing. But biologically and for your health, it turns out to be a very bad thing, because a lot of times when they do these hydrogenations, the resulting fats, instead of having a cis linkage like this, they end up with these trans linkages. And when they have these trans linkages, you can I don't have an explicit, they're not showing you that here, but you can, I think, see that if you had a trans linkage, these would be straighter, they would pack together better, and again, we're back to now a molecule that can perhaps form a solid at room temperature, but these are not natural fats, and that means that they don't get broken down in our bodies very well. And it turns out that many studies have shown that these actually tend to build up even more in our arteries and lead to atherosclerosis, right? And so that's why there's been a movement in recent years to sort of no trans fats, right? And so you'll see this now on a lot of packages in the store, no trans fats, and that's telling you that they are not introducing hydrogenated plant fats into that food, and so we shouldn't have the presence of any of these unnatural fats that are dangerous to our health. Okay, and then finally I want to say a little bit about phospholipids. So if we look at the structure of these triglyceride type molecules, we can have a situation like shown here, where we have, again, here's the glycerol backbone. And if we have a molecule that has two fatty acids linked to two of those, esterified to two of the hydroxyl groups on the glycerol, and in the third hydroxyl group contains a phosphate, this is called a phospholipid. And these are incredibly important because they also have chemical properties that lend themselves to forming biological membranes. And the reason for that is shown sort of in these illustrations right here. So the phosphate group is very charged, right? So this will actually tend to be, in general, sort of hydrophilic. It will like to interact with water. It has the ability to hydrogen bond with water, whereas these fatty acid linkages here are hydrophobic, right? Just as we just discussed. And so these will tend to want to avoid water. And so we end up with a molecule that looks like this, where we've got this hydrophilic phosphate head group on one end, and then the hydrophobic tails are hanging down in the opposite direction. And so these molecules you can sort of envision, and they have some nice pictures of this in the book. These can come together and interact non-covalently, where the hydrophobic tails are all lined up in one direction, and the hydrophilic head groups are all lined up in the other. And so if that happens in an aqueous environment, then we often get what's called a lipid bilayer, where we have these molecules lined up in one direction, and then a set of tails from another alignment of phospholipids coming together in the other direction. So we have head groups on one side and the other with these fatty acids in the middle, right? A very nice arrangement for membranes, because that means that the cell can use this to have the head groups interacting with the aqueous environment both inside and outside of the cell, with the fatty acid chains coming together in the middle and forming a very nice barrier for the cell, right? So these are incredibly useful, and we'll talk more about this when we talk about the structure of the cell, but we're going to see phospholipids coming up a lot in biology because they're so useful for forming membranes. And then finally, I want to say a little bit about steroids. So these are lipids that are characterized by a carbon skeleton that has 4 fused rings. And so this is an example of a steroid. This is called cholesterol. It's a very important steroid that is a component of animal cell membranes, and so is actually an essential component in our bodies. We hear about cholesterol a lot as being something bad, but we actually have to have it to form normal functional membranes in our cells. And the problem with it is that if we have high levels of cholesterol, we know that that correlates with cardiovascular disease, again with sort of processes that include atherosclerotic plaque formation, right? And so it tends to be the case that if a person has high circulating concentrations of triglycerides in the blood, so high amounts of fat, that also correlates with high amounts of cholesterol, and that can lead to buildup of plaques that we don't want, that can lead to blockage in blood vessels. So a couple of things about cholesterol and steroids like this. So you should sort of know, I think it would be useful to know this general ring structure, right? And what's important here is that this is the core of a steroid. So if you see this ring structure, this 4-ring structure, it's very characteristic, and you'll know that is some sort of a steroid. And you might not know exactly which one it was, because these steroids can differ by the functional groups that are connected to these 4 rings. But in general, if you see that set of 4-ring structure, you'll know this is a steroid. And these steroids are used as the building blocks for very important hormones, like sex hormones. Testosterone, estrogen, progesterone, other kinds of hormones are built from this basic starting structure. So we have to have cholesterol to make these hormones, and in the human body, cholesterol is actually synthesized in the liver. So we can get it both from our diet, but we can also get it through the natural biosynthetic processes that occur in the liver. And then some animals can actually, differs for different kinds of animals. Some animals can't make cholesterol, and they have to get all of their building block steroids from their diet, whereas others like us can both make it themselves and consume it and use it from their diet. Okay, and now, so I want to move on now to proteins. This is the third type of large macromolecule in the cell. And this is obviously an incredibly important class. They all are, but proteins are important in part because they're incredibly abundant. Your book says about 50% of the dry mass of most cells is accounted for by proteins. So these are really ubiquitous in cells. And they do all sorts of things. So proteins are important for supporting the structure of the cell and the kinds of macrostructures that are formed by cells, like organs and organelles, organs and whole organisms. They're important for storage of energy and storage of other kinds of useful building blocks for the cell. They help to transport various kinds of molecules into and out of cells and within cells to the places where they're needed. They're important for intercellular communication, so helping cells to understand their environment and what are needs that are coming up due to environmental changes. They help cells to move, and they also, of course, help cells to defend against foreign invaders and foreign substances. These are proteins. What are they? So they are, again, they are polymers, and we typically call them polypeptides. That would be a term that would be accurate for a protein, although for proteins that, for polypeptides that get longer than about 100 amino acid units, so these are polymers of amino acids, as we said, right? So when these polymers get longer than about 100 amino acid units, then we typically will call them a protein, whereas for a polymer less than 100 amino acids, by convention, we'll often call them a polypeptide. And that's rough, but that's sort of approximately the way those terms are used. And these proteins or polypeptides, these are built from the same set of 20 amino acids. And interestingly, this is true across all of biology. So, biologists have gone far and wide looking for wild and crazy organisms, and you can send Alvin down to, the depths of the ocean and collect bacteria that are able to grow at incredibly high temperature and pressure from these underwater geysers that are pumping out, water at incredibly high pressure, high temperature and under very high pressure. And you can look in those organisms, and then you can look at organisms that grow in the Arctic. You can look at organisms that grow in the Caribbean and anywhere else on planet Earth, plants, animals. They all have proteins that are built from the same 20 amino acids. Really interesting, right? So different kinds of proteins. And if you start doing the math, you quickly realize that 20 amino acids actually gives you an incredibly huge number of potential polymers, right? Because if we imagine that at each position in the chain we have 20 possibilities, you very quickly get up to a very large number. And that, in fact, biology doesn't even come close to exploring all of the possible polypeptides that would be possible based on 20 amino acids, because there isn't enough mass probably in the universe to do that, actually, right? So it's a huge number of potential proteins. And then it's important to know that a protein, if we use the word protein, this refers to a molecule that includes one, but it could include more than one polypeptide chains. So we could, in other words, we could have more than one polymer that fold together to form a functional protein. And we'll see probably at least several examples of that in this course, but it's very common in biology to have a functional protein that includes more than one polypeptide chain. And this is just a table taken from your book. It's meant to give you a few examples of the different kinds of functional roles that proteins can play. You don't need to memorize this, but it's important to know that proteins have this incredible diversity of function. And we're going to talk a lot more about this in a couple of lectures, but proteins play the role of catalysts in biological systems. In other words, they are the way that cells enhance the rate of chemical reactions to a meaningful rate. If we had no enzymes, none of us would be here, for sure, because the chemistry required for life, in general, occurs too slowly without enhancing those rates of reaction by catalysts. And proteins play that catalytic function in cells in general, right? So that's one important role of proteins, but it's certainly not the only one. So proteins also are important for structure, and they give you a few examples over here. Silk fibers, hair, nails, other kinds of structural properties are conferred by polypeptides or proteins. They can function as storage molecules, so to store, they give you the example here of amino acids, but they can store, they can also bind to other small molecules and store them for cells to use later. They help transport molecules into and out of cells and also within cells, so proteins will play that very critical role. And then, of course, they give you a few other examples here of proteins that can function as receptors. They can function as hormones. There are small peptides that are actually functional as hormones in the cell for signaling in different pathways in the cell. So incredibly diverse function. So naturally, there's been a lot of interest in proteins and how they're put together, how they're made, how they're structured. I wanted today really just introduce the basics of this. So this is a chart from the book that shows the properties of the amino acids or basic property, and then the 20 different versions of amino acids that we find most commonly in biology. So amino acids, so these are organic compounds that have a carboxyl group on one end and an amino group on the other side, and they're linked by a carbon in the middle. So this carbon is called the alpha carbon. I wonder if I should write that. Do I need to write that? It's in your book. You guys can write that down, right? So that arrow is pointing to the alpha carbon. And so this is the carbon that's kind of at the center of the amino acid, to which the amino group and the carboxyl group are linked. And then there's also, very importantly, in addition to a hydrogen here, what's called an R group. And the property of each amino acid is different depending on what that R group is. And so that's where the 20 different kinds of amino acids comes from. It depends on the type of R group that's linked to that alpha carbon. And those 20 different groups are shown in these charts here, and you can see that they've grouped them roughly by property. So these amino acids tend to have nonpolar R groups, right? So they tend to be more hydrophobic. These amino acids here tend to have polar R groups, so they tend to be more hydrophilic. And then these amino acids down here they can be charged, and so they can be acidic, as shown for aspartic acid and glutamic acid here, or they can be basic, as shown for this set of three, lysine, arginine, and histidine over here. And I don't know, Mike, where's Mike? Yeah, so do they need to memorize the 20 amino acids? How have you done this in the past? Right, that's what I would say too. Okay, I just thought I better check with the boss. But okay, so I think what we want you to know is, I don't expect you to memorize the chemical formula for all of these, but I do want you to know that there are 20 amino acids. I want you to know the basic structure of an amino acid. So if I showed you an amino acid and said, is this a lipid? Is it an amino, what is it? You would know it's an amino acid. And you would be able to point out what the different functional groups are. And if I showed you a particular amino acid and asked you what would the likely property of this one be, you'd be able to tell, because you could look at the R group and figure out, is this likely to be charged or not? Okay, does that make sense? And why do we care about these amino acids? Well, it turns out that when we link them together in polymers, they create properties in the resulting proteins that are incredibly important and allow these proteins to have all sorts of different functions. And of course, if they have different functions, that immediately tells us, if you're thinking as a sort of a chemist, right, that they probably have different molecular structures as well, and that's very true. So proteins can have a huge variety of different molecular structures. Let's see. And okay, but before we get to that, I want to just let's look at this. So we looked at amino acids; those are the building blocks. How do they come together to form polypeptides? Now, this is also something that you really do need to know. So I do want you to know this. Okay, I want you to know how individual amino acids come together to form a peptide bond. Those are the bonds that link together those individual units, and that's shown right here. So a peptide bond is this bond right here. It's between the carbonyl group on the end of one amino acid that's covalently linked to the amino group on the end of another amino acid. Okay, and there's a number of important consequences to this that I want to point out. So first of all, a peptide bond forms by this reaction here. We saw this earlier for carbohydrates. Does anybody remember what was that called? Right, dehydration, condensation, right? So we're losing water, same kind of chemistry going on. Even though the building blocks are different, we get loss of water and we get formation of a peptide bond. Now you know that this chemistry is reversible, right? So these bonds can also break. Does anybody know what happens when we break one of these bonds? What's that called? Hydrolysis. Yeah, right. So water is coming, did I say that right? Yeah. And right, so these bonds can break in a process in which we add water, and it's called hydrolysis. Now, when these get linked together, look what happens. We end up with a chain, a polymer chain, where the amino acids have a directionality to them, right? So we're going to end up with a polymer that has, on one end, an amino group right here, and on the other end, it has a carboxyl group right here. Maybe I should point to this chain right here. So we have an amino group on one end, and then we have a carboxyl group on the other end. And so in protein, and when we talk about proteins, if we talk anything about the structure or chemistry of a protein, or the way that the polymer chain is linked together, we're going to always be talking about, or we often will refer to the N-terminal end, the amino terminal end, or the C-terminal or carboxyl terminal end. Okay, so you do need to know that. That's very important to know, that there's a directionality to the polypeptide chain. And it comes about because these peptide bonds are always formed in the same way between the individual amino acids to form a chain like this. And you can see that this chain could be, this could be it. Maybe the polypeptide stops right here with 3 units, but it could be, thousands of amino acids long, right? And there are proteins in biology that are thousands of amino acids long. So these can be potentially very large. And so a couple more things about proteins. So proteins, these polypeptides that come together, either as a protein that might have a single polypeptide, or as we said, a protein could have multiple polypeptide chains to form the active protein. These have structures associated with them that give them very specific properties. So this is an example of an enzyme called lysozyme. This is a very abundant enzyme in our saliva, tears, our sweat. So what is lysozyme there for? It's actually a very useful enzyme. It helps to defend against bacterial infection because this is an enzyme that is able to bind to the carbohydrate molecules that are found uniquely on the surface of bacterial cells and cleave them, right? So it helps destroy bacteria that might otherwise infect us. And so it's an abundant enzyme. It's been studied for a long time by biologists and biochemists, and we now know that it has a polypeptide, the polypeptide forms a structure that's shown here, and they show you a couple of different ways that we often represent protein structures, either as a ribbon diagram like this, in which we don't see the individual amino acids in this polypeptide, but we see the shape that the chain forms. And then over here on the right-hand side is a space-filling model of the same enzyme. It kind of looks different, but I sort of like actually these space-filling models because it helps you to think about the protein as a real 3-dimensional structure that has functional components in that structure. In lysozyme, there's a very obvious groove that forms on the surface of this protein. It turns out that's exactly where these bacterial carbohydrates will bind and get cleaved. So just knowing something about the structure of this protein tells us a lot about the way it works. How do we think about protein structure? It's useful to think about it in terms of 4 levels of structure that are shown here. Primary structure is the sequence of amino acids in the chain. The secondary structure are the units of structure that form between a couple of chains, a couple of segments of that polypeptide chain. And in proteins, these are called alpha helices and beta pleated sheets. We will talk more about that in a future lecture, but just so you know kind of what's coming. And then those units of structure within the polypeptide chain can fold up together to form what's called the tertiary structure of the protein. That's what we saw for the lysozyme, for example. And then sometimes several different polypeptides will fold together to form a much larger protein that has what we call quaternary structure. And then finally, this is again just a couple of examples. Proteins can have a globular fold. They can also have an extended structure, as in the structure of collagen, which is a skin protein. And finally, how do we understand protein structure? You might be thinking, how do we know what these proteins look like? It turns out that it's possible to crystallize proteins, and we can shoot X-rays at them, and from the diffracted X-rays, calculate a diffraction pattern that is consistent with only one type of 3-dimensional structure in that crystal. And this is the technique that's been used very commonly, really since the 1960s, to study protein structure and determine structures in detail. So lots of the beautiful structures you'll see in your book and that we'll talk about are determined using X-ray crystallography. Okay, and we have 5 minutes left. We're just going to say very briefly, talk about nucleic acids. Nucleic acids are very near and dear to my own heart, so I'm an RNA biochemist. I've been studying the structure and function of RNA since I was in graduate school. And so why do we care about nucleic acids? Well, you probably know that this is the genetic material. We have 2 flavors of nucleic acids, DNA, which stands for deoxyribonucleic acid, and RNA, which is ribonucleic acid. You might think, well, they're not that different from each other, and we'll see chemically that at a chemical level they don't look that different, but biologically they have very different kinds of behaviors and properties. This is just, again, we don't have to go through this in detail. You can look at this figure in your book. This is what I like to call the central dogma that shows you that genetic information passes from DNA to an RNA copy and then is eventually read in the cell by the ribosome, which makes a protein molecule based on the genetic material encoded, genetic information that's encoded in DNA or sometimes actually in RNA, right? So RNA can also be the primary genetic material in viruses, for example, like the AIDS virus is an RNA virus. Influenza, flu, is also an RNA virus. So RNA can be a primary genetic material as well. And so these are also polymers. They're made of monomers that are called nucleotides. And each nucleotide consists of a nitrogenous base, a pentose sugar. So pentose, we talked about that earlier. That means it's a 5-carbon sugar, right? And a phosphate group. So those are the 3 components of each nucleotide. If the nucleotide lacks the phosphate group, then it's called a nucleoside. So you'll see that sometimes in your book too. So that's the sort of the definition of those terms. And here's sort of a useful diagram that shows the arrangement of these individual nucleotide building blocks with the phosphate group connected to one of the carbons on this pentose sugar, the nitrogenous base connected to another carbon on this sugar, and depending on this is very important, small difference, but it's really important. So depending on whether we have a hydrogen here on this 2' carbon of the pentose ring of the sugar, if we have a hydrogen here, this is a deoxyribose, and this is the sugar found in DNA. if we have a hydroxyl group at that carbon, this is a ribose, and then it's sugar that's found in RNA. And if you take biochemistry later in your career here at Berkeley, you'll certainly hear a lot more about why that's so important structurally. But for now, it's just important to know that difference between the sugars in DNA and RNA. One more thing. So these are the nitrogenous bases. These are the purines. These have two rings associated with them, adenine and guanine. And these are the pyrimidines. In RNA, this version of pyrimidine, this version called uracil, is found. In DNA, there's no uracil, but instead, it's thymine. And if you look carefully at those structures, you can see these two differ by just one methyl group right here, right? That is an important difference between DNA and RNA, and this is an important difference between DNA, RNA in the backbone. All right, so we'll talk more later on Monday about this, and we'll start talking about the structure of the cell. Have a nice weekend.